lecture, and today we're going to be talking about chapter 16 in your book. And this chapter is talking about the molecular basis of inheritance. So we're going to talk about how um, does inheritance happen through the um, through the DNA, right? Mostly. So we're going to see how DNA is basically copied during transcription. And as many of you know, that the transcription happens, right, in the nucleus. Translation happens in the ribosomes, okay? As we kind of went over that in the first few chapters when we talked about the cell. Now, the DNA is also replicated during mitosis, right, and meiosis. So it has to be basically copied um, to be able to basically let the cell go through cell division and basically give out daughter cells. Okay, so let's start going with this PowerPoint and talk about this lecture slide at a time. Okay, so what do we know so far about the DNA? So we know that it's double helix, right? So it's got basically two ribbon-like structures, right? And these are the ribose, right? They're sugars based, and attached to those sugar molecules or ribose, there are also phosphate groups, okay, and there are also nucleotide bases, right, that pair up and attach the two double helix together. Okay, so the nucleotide bases, we will go over them in a bit, right? But just to give you a general idea, there are four of them in the DNA. So we're gonna just call them, A. We're gonna call the first one A, the second one T, third one is C, and then the fourth one is G. Okay. Now I'm gonna talk to you, how each one pairs with the second with a specific nucleotide in the next few slides. But I'm just giving you a general idea before we dive into the lecture. Okay. So the DNA replication, as we know by now, happens where? It happens during the S phase, right? And it's gonna happen in the nucleus, right? Before mitosis starts, and basically mitosis is gonna give out the two daughter cells at the end. So let's see what's going on here. So DNA replication, right, allows for genetic information to be inherited from the parent cell, right, which is basically that cell that's maturing, right, and it's going through the S phase, and then eventually it's going to go through mitosis, right, as we know by now, the M phase, and that's going to give us the two daughter cells at the end. Okay, now that is in mitosis. In meiosis, right, as we know by now, the final daughter cells, we're going to get four daughter cells, right, from a parental cell. So because meiosis involves meiosis 1 and meiosis 2, right? And so the four daughter cells in meiosis 1 actually have the half of the genetic material as the parental cell, unlike the mitosis, okay? So that's why we call them haploid, right? In the case of meiosis, okay? So here you're seeing how basically you have the DNA that's going like in an uncondensed form into a maturing cell, right? And then that unduplicated DNA, right, in the form of a chromosome, it's gonna basically be duplicated in the S phase, right? So that is what we call basically the DNA replication during the S phase. Okay, so before we go and talk about that, just know that when we say a gene, right, gene is a unit of heredity that contains the basically multiple specific nucleotide bases, and they're basically fragments of a DNA. Okay, so we could say that for example, a specific gene, like for example for eye color, is located at that location of this chromosome. So we're gonna call this what? A locus, right? So it's talking about a specific location. We went over that in the last few chapters, okay? So, excuse my voice today. So basically, the before the DNA proceeds to replicate, it's gonna form those little bubbles, right? And those bubbles are gonna basically proceed to form the newly formed strands, right, of DNA. Because remember, we have double helix, so basically we have to split the two strands, okay, and then from each strand here, which is what we call the parental strand or the template strand, okay, we're gonna make a newer one on each side, okay. So eventually at the end we're gonna basically get two sister chromatids. Do you remember that? Okay, so that is what's happening during the S phase. Okay, so you see how you have the original parental or what we call also template strand, and you can see the light blue here. That is the newly formed DNA that we replicated in each sister chromatid. Okay, so during the 20th century, there were many biologists and scientists, right? They were trying to figure out like how does information get passed on from from parents to offsprings and things like that. So they wanted to know what kind of molecules were involved. Okay, so they had in mind two molecules that they were looking at, and the first one was proteins. Okay, as we know what they are by now, right? And then the second one was DNA. Okay, I wanted to find out by doing experiments which one actually carries the genetic information, genetic material to actually have a manifested or a expressed phenotype in the offspring. Okay, so let's keep going and see what happened. Okay, so T.H. Morgan, that is, he was a scientist. Okay, so he was working with a group, right, and they showed that genes are located on chromosomes. I think we saw that in the last few chapters, right? They recognized that genes are located at specific locations, and they formed a map of those genes, right? Um, so they also figured out that the two components of chromosomes, right, DNA and protein, they became the candidates for genetic material, as I told you, right? So they had in mind these two molecules, and they were trying to figure out, hey, which one is actually responsible for the genetic material to be inherited, okay? Okay, so they found out, by doing an experiment, specific experiment on actually infecting bacterias with the viruses, okay? So we know, right, from our knowledge that viruses and bacteria infect humans, right, in different ways, and they're two completely different microorganisms, right, that are treated also differently, right? Bacteria we can treat it with antibiotics, while viruses we can't, right? Because of the nature, of how their anatomy is. Okay, so let's dive into the actual experiment that gave them ideas what's going on here. Okay, so um, that experiment also kind of started with Frederick Griffith, right, in the 1928, and so this gentleman worked with basically bacterias and viruses. Okay, and what he did is he worked with two strains of bacteria, right? So one was basically pathogenic, or it's known to lead to death, okay, of the organism, and the other strain of bacteria was known to be non-pathogenic, or that could be also used as the control, right? So this strain was known that it's not gonna basically lead to the death of the organism. Okay, so this scientist did, okay, so he infected first just to make sure that we're getting the results that needs to be gotten here. Okay, so these two are just confirmation of the actual bacteria. Okay, so between the two kinds. So he infected the first one with the pathogenic type of bacteria strain, and that led to the mouse, dying. Okay, so that is a confirmation of what's going on here. Okay, now the non-pathogenic bacterial strand gave a healthy mouse, so that is expected. Okay, so these are both controls, right? We're trying to basically establish that these strands actually lead to the known information about them. Okay, now what this gentleman did, he actually killed the um, the uh, the pathogenic bacterial strand, okay, using the heat as a source. Okay, so he heat killed the S cells, okay, and so the S cells are basically the ones that are basically infected. Okay, so when he did that, okay, he noticed that the okay, I'm sorry, I'm gonna reverse this a little bit. Okay, so he'd killed the S cells that are non-pathogenic control. Okay, so he he um, he did that on those non-pathogenic control. Okay, so which led to the mouse being healthy. Okay, now after that he mixed, okay, the ones that were killed, so the ones that were the living S cells that were killed by heat. Okay, okay. So remember that he killed the the cells. Okay, but however the genetic material are still there. Okay, so he mixed then the S cells and the living R cells, okay, which are basically non-pathogenic. He mixed them together, as you can see here, okay, and injected it into the mouse. And so what he noticed was that um, the mouse actually died. Okay, although the S cells were heat treated, but the mouse still died. Okay, so that is because there was some type of what genetic material transfer, right, that happened between the heat killed S cells and the living R cells. Okay, so basically living the living S cells, okay, um, basically were alive because because of the transfer of the genetic material. They start being alive, and so when that happened, the mouse died. Okay. So he called this whole phenomenon a transformation. Okay, so that is a change in the genotype and phenotype due to assimilation of foreign DNA. So that means, okay, that the DNA of the original cell got transformed, okay, due to a assimilation, or basically being in contact or being close to foreign DNA, which in this case it would be what the heat-killed S cells, which are known to be pathogenic. Okay, so these S cells were responsible for the result that we see here. Okay. Okay, so more scientists obviously worked on this to try to identify, if the DNA is in fact the molecule that is actually transferring the genetic material over. Okay, so these scientists listed here, Oswald Avery, right, and Macklin McCarty and Colin McLeod, right, all of these identified the transforming substance as DNA. So again, they were trying to see, is DNA actually the actual answer to all of this, or is it proteins? Okay. So again, you're seeing, on this, that many biologists were still skeptical about DNA. They didn't know all the information we know today. Okay, so let's see here. So they again started studying back the how viruses infects bacteria. Okay, so that is how they gather more evidence that DNA was the genetic material that basically allowed the viruses to basically infect and inject their genetic material into the bacterias. Okay, so we call these viruses, right? We call them bacteriophages. Okay, so they're gonna basically hijack a bacteria. Okay, so a virus normally it can be DNA or RNA. Okay, so it doesn't have to be DNA. Okay, so it just depends on the virus. For example, HIV is a retrovirus, or an RNA. Okay, so that viruses normally are enclosed by a protective coat, as you can see here. Okay, and then basically they have their DNA inside the head. Okay, so what they do is basically they hop on onto a bacterial cell. Okay, and they attach to the membrane of that cell, and then they basically inject their genetic material, okay, through their tail. Okay, so after they do that, basically the genetic material will basically travel into the cell, okay, whether it's a bacteria or whatever, or any type of cell, and then it will basically enter if the DNA is inside the nucleus, it will enter the nucleus, and then it will basically hijack the DNA and start basically taking over the machinery of the cell, okay, and start making copies of itself. And that's how the virus basically hijacks and is able to actually reproduce through the basically whatever cell that the virus is hijacking. Okay. Okay, so in 1952, Alfred Hershey and Martha Chase, right, they showed that DNA is the genetic material of a phage known as T2. So that is T2 was the one they were working with, and that is a type of virus. Okay, so let's see what kind of experiment they designed, right? So they were working with T2, okay, um, and they showed that one of the two components of T2, right, either DNA or protein, enters the E. coli. So E. coli is what? It's a bacteria. Okay, so we're working with the same thing. T2 is a virus, E. coli is a bacteria, and they basically were watching what happened during an infection. Okay, they were trying to see and differentiate is the DNA is the one that's basically being injected uh, by T2 into the E. coli, or is it protein? And you know, as a result, we're getting the basically reproduction, or you know, the virus is able to basically reproduce themselves in the host cell. Okay, so they concluded, right. So the results showed, and we will look at the experiment in a little bit. So they concluded that the injected DNA, okay, of the phage, that's the one that is actually providing the genetic information. So let's look at the actual experiment here. So the first one here on top, okay, is basically involving looking at protein. And then the second one, which is batch 2 here, down here, it's basically the one that's involving DNA, okay? So what they did was they were working with E. coli bacteria, so that is the bacteria right there that you're seeing. And then the phages, or the viruses, okay, these are the ones that are here on top. Do you see them, the pink ones? Okay, so they did the same thing here and there, right? And what they did is that they used a radioactive uh, sulfur, okay, which is basically to identify proteins. So if the material is passed on, you're gonna basically get at the end what? A pink result, right? Pink stained result, okay? If in the case of the DNA batch, okay, they use radioactive phosphorus, and that is basically blue normally, okay? That's how it looks like. So they injected those in the viruses, okay, and they were watching to see when the viruses, basically try to infect the bacteria here, okay, they basically mix them by centrifuging them. Okay, so they allowed the phage or the viruses, T2 viruses, to basically get basically and infect the bacteria, okay, that is involved with, by mixing them, and then they centrifuge them later on to basically that's after the infection happens, as you can see here, okay. The centrifuging will basically allow the viruses to actually go away from the bacteria, okay. So you're left with an actual bacterial cell by itself, okay, and the viruses go away, all their material. So what they saw was that that's the most important part that we're concerned with here, okay. So what they noticed is that the genetic material actually was passed on. Do you see how it's blue here? Okay, so that means the DNA that was inside the viruses, the T2 virus, actually got into the bacteria. Well, if we look at the protein, that is not pink. Do you see that? Okay, so that means something else. Okay, that most likely it's the DNA here, right? But in their case, the pink that was stained did not actually go in, so that means what? That proteins are not the ones that are basically involved in the transfer of genetic information, okay? However, DNA is, as you can see here, okay? So, let's look at the Hershey and Hershey-Chase experiment. They were basically involved with this experiment here. You can see, if you want, you can basically play the videos and look at the whole thing on your own time. Okay, so let's keep going with this. So let's talk about a little bit the DNA structure here. Okay, so I gave you a little bit of introduction early on. Okay, so again, you're seeing here one of the helix, right, of DNA. Okay, so again, that ribbon-like structure, that is what we call the ribose, and it's got basically phosphates on it, okay, phosphate group, okay, and then you have the nucleotide bases, okay, and we said that there are 4 of them, right? So we have the T, G, C, and A, okay. So the way this works is that normally if there was another strand here, right, because there's 2 strands in the DNA, it's double helix. So normally the way this works is that thymine will bind with adenine. So T goes with A always, and then basically guanine will always go with C. Okay, so you can imagine if this guanine wanted to bind to something, it's going to bind only to cytosine or C. Okay, and thymine will always bind to adenine. Okay, so let's see here. So now, basically, scientists, started to realize that, hey, DNA is the one that's responsible for the manifesting or the expression of genes, and that's what they were saying, right, with all those experiments, okay, not proteins, okay. Okay. So that is, they were looking at basically studying those nucleotide bases, okay, and Chargaff's, right, also a scientist, found out that noticed that there are basically similar numbers of basically specific nucleotides, which is these 4 in the DNA they were looking at. Okay, so that means they found out, for example, that um, let's say, I'm just giving you I don't remember exactly the numbers that they were looking at, but let's say just give it a rough estimate. They found out, for example, that the DNA molecule contains 300 thymine, okay, and 300 adenine, while there was 200 guanine and 200 cytosine. Okay, so from that they learned that G binds with C because of similarity in numbers. Okay, so and T binds with A. Okay, that is how they found out, initially, how all of these things bind together. Okay. Okay, so after DNA was accepted, right? Now they accepted that, hey, it's a DNA, it's not the protein that's responsible for genetic material, right? Now they wanted to see how does actually the DNA structure, right, which is the double helix structure, and basically, it involves chromosomes, right? As we know, all of that. It involves basically genes that are basically segments of that DNA and all of that. How does this account for the heredity, right? Uh, how is it responsible for passing on genetic information? Okay, so Rosalind Franklin and Maurice Wilkin. Okay, so they use a technique. It's called X-ray crystallography. Okay, so they basically did an X-ray, as you can see here, and they noticed the double helix of the DNA. Okay, you can see it right here. Okay, so let's see here. So they noticed that the DNA was helical. Okay, so they also were able to figure out the spacing between the nitrogenous bases. Okay, so now they know that from previous experiments, as we saw, that there was 4 bases, and that the A binds with T, C binds with G. Okay, and now they start knowing that, hey, it's a double helix, and that the nucleotides will basically they will find out that they pair with each other between the double helix. Okay, so that is what you're seeing here again. Okay, you got the deoxyribose sugar right there. Okay, and then you have the phosphate groups. There's nothing new here, right? We've been talking about this since the beginning of this chapter. Okay, you got the nucleotide bases. T binds with A, as we know. G binds with C. Okay, that is one strand, and that is your second strand right there. Okay, and what you notice here that they are what we call anti-parallel. Okay, so the 5 end is to the 3 end, and then the second helix binds to it the opposite, 3 end to the 5 end. Okay, that is how DNA works, and because of that, it's able to form this double helical structure. Okay, so basically, some scientists they were trying to figure out, which base, nucleotide base pairs with which other one. Okay, so they tried to do different ones like A with A and A with G and so on. Okay, and basically, they found out that not everything works, right? So, purine with purine was too wide here, right? And pyrimidine with the pyrimidine was too narrow, okay? And then basically they finally figured out that purines and pyrimidines, so basically adenine and, sorry about that, so purines and or adenine and thymine, okay, they're gonna basically have a consistent length, right, between them, and that is consistent with the X-ray data that you saw here, okay? Okay, so that is also showing you, the same thing I told you about the Chargaff's rule, right? That in any organism, the amount of A is equal to T. Okay, why? Because, they have to bind to each other. So you can't have like more of A's than T's. Okay, and then same thing with guanine and cytosine. Okay. Okay, so now we're gonna basically dive into the actual DNA replication. How does it happen? Okay, we know that it happens in the S phase. Okay, and we know that it happens before mitosis and before meiosis start. Okay, so mitosis and meiosis will actually give you the offsprings. Okay, so let's see here. We know we're going to end up with 2 daughter cells in the case of mitosis and 4 daughter cells in the case of meiosis, right? So the copying of DNA, it's called DNA replication. We know this happens in the S phase, hopefully. You know this by now, right? Okay. So, and we went over this a little bit in the beginning, that because there's two strands, okay, the way when DNA replicates, right, it's gonna be split up as you can see here, okay, by an enzyme. We'll see how that works in a bit, okay. So the 5 to the 3 end will split away from the 3 to the 5 end strands, okay, and each one becomes what we call parental or template strand, okay, and then a newly formed um, strand will be added on to that parental strand, okay. Eventually what we're gonna see is that the complete complete DNA is gonna be formed into these sister chromatids as we saw earlier. Okay, so let's talk about the actual DNA replication. So that is your parental molecule before it starts, before it splits up, basically, right? This is after it splits up. Okay, there's nothing like too crazy about this. Okay, so that is the double helix, and it splits up using an enzyme. We will see how that happens. Okay, and each one of them, they're gonna become the template or parental strand. Okay, here's the light blue newly formed strand now. Okay, you will notice that the bases are complementary, right? They have to match, otherwise this is not going to work. So the newly formed strand is going to have, a nucleotide base that is, like, for example, a T is going to go with an A, G is going to go with a C, and so on. That's how these strands get added. And you can see that both strands have, a newly formed complementary strand. Okay, that is how we're making our newly formed copy of that parental molecule that we started with. Okay, so the scientists here were coming up with different models of how the actual strands actually split up during replication, and how do we get the final result here. Okay, so the final result that the scientists actually agreed with, that this is what's happening, is called the semi-conservative model, and we will see basically all the different models that show what each one does, okay, and why they picked the semi-conservative model. Okay, so this one predicts that when a double helix replicates, each daughter molecule. Okay, so let's look at it. So this is your semi-conservative molecule. That is the original DNA here, the parental DNA. Okay, you can see that when um, the strands are separated by the enzyme, okay, that is your dark blue parental cell or template cell, or not cell, sorry, strand. Okay, you can see how it gets split up here and there, okay, and how basically each strand is basically getting a newly formed complementary strand. You see that? And then eventually, when the final, so this is your final replication S phase, and then if those cells go through DNA replication again, this is how basically we're splitting up the, the genetic information from the original parental cell. Okay, so the competing models were called the conservative model, which is this one right there, and then the dispersive model. So let's see what each one is. So with the conservative model, it's in the name. This is saying that the parent cell is gonna conserve its DNA. Basically, it's like passing on the whole thing, okay, and making like a completely newer, sorry about that, new strands. Okay, you can see that. But from what we know so far, that's not true, right? We know that the strands get actually split up, and each strand copies basically itself, as we saw previously. Okay. Now the dispersive model is kind of saying that the basically fragments of the strands are basically uh, conserved in the newly formed daughter cells, and while other fragments of basically newly formed DNA fragments are added to the original parental fragments. Okay, and that is not true either, right? But we do not see this during DNA replication. Okay, so that is why the semi-conservative model is the one that basically Watson and Crick's basically agreed with. Okay, so let's take a closer look at the actual how does replication happens. Okay, so there is always a starting point for DNA replication. Okay, so we call this starting point the origin of replication. Okay, so it's in the name. Origin means the starting place of where the replication starts. Now, basically, the DNA, okay, is able to our DNA in eukaryotes, okay, is able to form several origin of replication, okay, uh, by making these what we call bubble. Okay, so each one of these strand is basically it's a strand of the DNA, right? So both of them will make the double helix, if you imagine that, okay? So the enzymes will basically create bubbles here and split up the DNA molecule in several sections along all the DNA, as you can see here. So that will basically allow this whole thing to be replicated at a much faster rate because of the several bubbles that are being basically created, and so replication can proceed at several places at the same time. Okay. So in bacteria, however, there is basically only one origin of replication. So only one place is where the replication will happen and start. In eukaryotes, we have several places, as you can see here. Okay. So that is showing you the bacteria here on the left side. You can see that it's only one origin of replication here. Okay. You can see how there's a replication fork, right? That goes in both ways. Same thing with eukaryotes. So this is eukaryotes. So we are a eukaryotic organism. Okay. So you can see that there are several origin of replications here, while there is only one here. Both of them will start the replication basically in both directions, okay, left and right. And then you have the two daughter cells at the end, regardless of whether it's a bacteria um, or eukaryotes. Okay, so now let's talk more like into detail of how the actual replication happens. Okay. So we have the double helix here. There's nothing new, right? We know that it's in the 5 to 3 and 3 to 5. That's how it is, right? It's antiparallel. So the enzyme helicase is the one that basically is gonna split up the two helix, as you can see here. So it splits them up. So every time you see basically a bubble here, there's that helicase enzyme uh, that is doing that, okay, on both sides. That's what these enzymes do, okay. Now you'll notice here that there's also some gray proteins that we call single strand binding proteins, and these proteins are going to be attaching to each of the um, split up um, strands, and what they're gonna do, they're gonna allow these strands to stay apart and not stick back together. They're gonna stabilize, okay, each of the parental strand. Okay. Now you'll see here that there is also topius merase. Okay. Uh, so I'm hoping that I'm pronouncing this correctly. Okay, topoisomerase. So that is another type of enzyme. Okay, so what this one will do is it will basically prepare the DNA. Okay, for helicase. Okay, so it's gonna relieve basically the strain of twisting. Right? You remember how the DNA, you know, molecule or structure of the DNA is like kind of like an uncondensed and like kind of like spaghetti form. Okay, so it's like squiggly and zigzag and it's all over the place. So that enzyme will basically help to alleviate the strains of twisting. Okay, to help heligase. Okay, so you will also notice here that there is another enzyme that enzyme is called primase. Okay, it's gonna basically get attached to the parental strand. Okay, and that primase is gonna basically create, okay, a RNA primer. So it's gonna make an RNA primer and then it's gonna put it in the 5 to 3 direction. So if it's working on a 5 to 3 DNA strand, it's gonna basically put it what? Anti-parallel, same thing like how this strand was on it. Okay, so that is basically given direction to the newly added uh, complementary bases. It's telling them that, hey, this is the primer, right? And we know what a primer does, right? It's like a prepare or a mark of foundation, right? It's gonna prepare for the new coat, if you will. Okay, we're gonna put primer here. Okay, and this is how this happens. So it proceeds by putting an RNA primer here, and then that's going to give direction for the replication fork to proceed from right to left. Okay. So let's now talk about the DNA polymerase. So DNA polymerase is the actual builder. Okay, that is what I call it the builder of the actually complementary newly formed DNA strand. Okay, that is what's gonna build it. So this one is gonna basically get the signal that, hey, there's an RNA primer here, and we gotta start building that newly complementary strand, okay, based on the RNA primer direction here. Okay. Okay. Now, let's see here. So again, the primer here, as you can see, is a short primer, right? It consists of only 5 to 10 nucleotide long. So that is basically just, again, it's a signal, okay, that, hey, we added those strands here. Now we can continue adding the strands that follows after that, okay? Or the nucleotide bases, I mean. Okay, so DNA polymerase, as I told you, is the one that basically is going to do the synthesis of DNA, okay? So the rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells, okay? So let's see here. So each nucleotide that we're going to add using the DNA polymerase is actually a nucleoside triphosphate, okay? So that is kind of what you see with what type of molecule? ATP, right? It's got basically the adenosine triphosphate, so similar structure here, okay? So the DNA ATP supplies adenine to DNA, and similar to ATP of energy metabolism, okay? So that is what I just told you, right? So it is similar to the ATP molecule, okay? So the difference is, guess what the difference is? That, what do we say? We say DNA, so DNA is deoxy, okay? Deoxyribose sugar in it, while ATP is actually a ribose. Do you remember that from the structure of ATP? So So there's actually difference in the sugar here between the two molecules, okay? So at each monomer, as each monomer joins the DNA strand, right, there's a dehydration reaction. So what is the dehydration reaction? Is splitting of ATP, right, into adenosine diphosphate and phosphate, okay? We'll see how that happens. So we're losing two phosphate groups as the molecule of pyrophosphate. So let's see how this happens. So I know this could be a little bit confusing because you've never seen it before, but here's the template strand, so dark blue, okay, and here's your newly formed strand, okay? So again, if we're going in the 5 to 3, we're gonna start building the newly formed strand in the 5 to 3 direction at all the time. If you get a question on the test that asks, you know, what direction is the replication happening, you're gonna answer always in the 5 to 3 direction, okay? 5 prime to 3 prime direction. So here is the incoming nucleotide, and what did we say it's made out of? It's made out of a deoxyribose, okay? And it's got 3 phosphates similar to the ATP molecule, right? So ATP has 3 phosphate groups. So remember, adenosine triphosphate, okay? Here's your deoxyribose sugar, okay? And then here is the nucleotide base attached to it. That is what makes it different than an ATP molecule, right? So you got this base being attached here, which could be not just adenine, but it could be thymine, it could be guanine or cytosine. Okay, and then you have a deoxyribose sugar. Okay, so the way this happens is using DNA polymerase, which is the builder of that strand. Okay, so the way this happens is we have two of the phosphates. Okay, do you see that? Basically, get or leave due to the hydrolysis that's happening here. Okay, you can see how they leave and break apart from the original molecule, and then one of the phosphate is only left, and that is basically what is made out of what DNA strand is made out of, right? It's only made out of one phosphate, one deoxyribose sugar, and the nucleotide base. Okay, that's what makes a DNA strand that we're looking at here. Okay, so that reaction is happening, that hydrolysis is happening every time DNA polymerase, which is the builder, is building a newly incoming uh, DATP molecule. Okay. So again, you know now that the DNA helix, both double helix, they're anti-parallel, right? So they're 5 prime to 3 prime, and and the 5 prime to 3 prime in the other direction. Okay. So I just told you this, literally 2 minutes ago, that DNA polymerase add nucleotide only to the free 3 end of a growing strand. Okay, which means the new DNA strand can elongate only in the 5 to 3 direction, as I told you. Okay. So this is talking about here when it says 3 end, it's the 3 end of who? The RNA primase, right? The primer. The RNA primer that was added by primase. The new nucleotides, they're going to start being added at the 3rd end of that primer. Okay, so here's your primer here. Okay, so you can imagine that these remember how I told you that each bubble, the replication is happening in both directions. Okay, so there's always that direction. So imagine helicase, the enzyme that splits those 2 strands. It's over here and over there. Okay, there's always a leading strand. Okay, which is basically where the actual leading replication is happening. Okay, you can see where the primer is here. Okay, so this side of that primer, okay, on the left side, it's gonna be the 3 prime end. Okay, and then basically we're proceeding in a what? A 5 to 3 direction. Okay, remember that. So we have the the DNA polymerase synthesizing in the the leading strand. Okay, now on the opposite side, there's also also replication happening, right? Because we want to complete the whole thing at the same time. Okay, so the other side of that fork on this side, you can see. Okay, this side we call it lagging strand. Okay, now in the lagging strand is a little bit different. Okay, because in the lagging strand we're gonna add a RNA primer in basically segments instead of a long like a long replication fragment like the leading strand. We're gonna add them here in small fragments. Okay, and in the lagging strand, we're going to call these fragments, small ones, Ogasaki fragments. Okay? So let's keep going and see what's going on here. So let's look at the first at the leading strand. Okay? So you look at this leading strand here. Okay? That is the 5 to 3 prime. Here is your RNA primer. We're adding to the 3 prime end of that RNA primer. Okay, we're using what? We're using DNA polymerase here to add and build the newly formed nucleotide bases. Okay, it's actually the whole, as we saw, the whole DATP molecule being added. Okay, we know that each one uses hydrolysis to add that molecule onto the onto the template, using the template but onto the newly formed strand. Okay, here is the lagging strand on this side. Okay, so the leading is going this way, the lagging is going this way. Okay, so let's keep going and see what happened. Okay, there's nothing new here. So I wanted to also mention something here, that DNA polymerase actually slides on the synthesis location or the replication, okay, by using a sliding clamp. So that clamp is gonna basically slide the DNA polymerase down into the 5 to 3 direction, and it's gonna allow the DNA polymerase to build fragments of that newly formed complementary strand one at a time. Okay. So again, we're going to talk about now the Ogasaki. I told you this just a little bit ago, right? So the elongation of the DNA strand, right, of the newly or complementary DNA strand is going to happen also by synthesizing, in the other direction, which is the lagging strand, as you can see here. Okay, to complete that whole bubble of DNA replication. Okay, so I told you that each fragment, okay, is going to be called Ogasaki. Okay, fragments. Now, if they're fragments, that means they have to be joined together to match, what's going on here in the leading strand. Okay, so that is where DNA ligase, it's another type of enzyme. I know there's a lot of enzymes, but take notes on what each one does. Okay, so DNA ligase is kind of like the super glue. Okay, so ligase is kind of like a super glue. Okay, so G for glue. So that is going to basically glue together and attach together the Ogasaki fragments. Okay, so that is showing you now the synthesis of the lagging strand. Again, this is in detail, more detail. This is what you're seeing here. So there is always an RNA primer. As you can see, there are several RNA primers that are happening at the same time. Okay, so the RNA primer basically is made by the primase, as always. Okay, then the replication is happening this way, right, in the 5 to 3 direction. Okay, so we are looking at this strand right there, not to get confused, okay? And then after this happens and elongation starts, another RNA primer, okay, is gonna be made as well. Okay, so it goes like basically from here to the to basically behind that, and then behind it, and so on. That's how we create those fragments. Okay, and basically every time we create a fragment, we're gonna create it using what? DNA polymerase, as always. Okay, there's nothing new here. So DNA polymerase will synthesize that fragment, be done with it, with the fragment, and basically detaches. Now, after that DNA detaches, another RNA primer is added to the back of that fragment that we created. Okay, here is that. See it? That's the one we created, and here is the newly added RNA primer to the back. Okay, and here is what DNA polymerase again with the sliding. Um, that is basically synthesizing that fragment as well. So once that is synthesized, and that already has been synthesized, more fragments are being synthesized, and who is attaching all of them together? DNA ligase. Okay, don't forget this point. Okay, so before we forget, who is replacing the RNA primers? So remember, we added the primers as a starting point, right? But we need to basically, replaces the RNA that was added, because that is a DNA molecule, right? So the one who does that is called DNA polymerase 1. So there's actually two types of DNA polymerase that you need to know. Okay, so just know that the builder, okay, both of them are building, by the way, but the one that is like basically elongating the strand is DNA polymerase 3. And the one that is actually also building, but only replacing RNA primers with DNA nucleotides, is called DNA polymerase 1. So the only thing that the difference is the number here. Okay, and we know that ligase, DNA ligase, is the one that's gonna basically glue together the fragments after the DNA polymerase 1 basically replaces the RNA bases with DNA ones. Okay? All of those videos are very good, very nice to watch. They're animated. Please take the time and watch them on your own. They're helpful, so... Okay, and that is basically how that's a summary of how the whole thing happens. So that is your leading strand with the sliding protein, and then you have the DNA polymerase 3, which is the builder. That is your lagging strand on the other side. You can see how it's being synthesized, in fragments. Okay. Here's your DNA ligase. How it's basically attaching all of those fragments together. Okay. And here's just a summary of what each enzyme does and the name of it. Okay. Okay, so let's see here. So we can see that the DNA polymerase, as you saw, is reeled in on the parental DNA strand, right? And that's how basically it adds new the newly complementary strand. Okay. So this is what you're kind of seeing here. Okay. So that's the unwinding, right, of DNA. That's just showing you kind of the same exact thing. Okay, so now that we know how replication happens, okay, and you might have probably remember from previous lectures that during any type of copying and pasting DNA, sometimes we can get a mismatch, right, due to any type of it could be a mutation, it could be any type of things that are going on while replication is happening. Okay, so um, if in this case a mismatch happens, which is where we add a incorrect nucleotide in a place where there is actually need to be another type of nucleotide. Okay, there are repair enzymes that are gonna correct those errors in the base pairing. Okay, so what kind of things that could allow these mismatch to happen and could increase the possibility of these things happening? Things like, in the environment, such as chemical, physical agents, okay, like cigarette smoking, X-rays, UV lights, right, from the sun. What else? Carcinogens, right? A lot of these things will basically uh, affect the DNA replication and possibly cause a mutation. Okay, so the repair happens using basically a nuclease enzyme. So as you can see here, so there's a mismatch that happened here. Okay, nuclease enzyme will come in here and basically uh, do a nucleotide excision repair. Okay, so it's gonna cut out the badly formed, the mismatch basically of the badly formed nucleotides, cut it out, and then after they're out, their DNA polymerase, which is the builder, okay, it's gonna come in and rebuild the correct nucleotide bases, and basically DNA ligase is gonna glue things together, as we know. Okay, so that is how this thing happened at the same time. Okay, so these changes, right? We call them mutations. Okay, sometimes they happen due to many reasons, as we saw. Okay. All right, so this is just also telling us that, replication machinery, okay, will always go in the 5 to 3 direction, okay? Let's see here. Okay, so also many replication of the same, so let's say a parental cell is replicating its DNA and going into the two daughter cells. Those two daughter cells are going to do the same thing and go into the other two daughter cells. Eventually, every time the cell replicates its DNA, okay, it's gonna basically uh, create shorter DNA fragments as they go. So repeated rounds of replication produce shorter DNA molecules with uneven ends. Okay, so that is in the case of eukaryotes. In the case of bacteria, because their DNA is circular, this does not happen. Okay. So here's what's happening when replication, when like cellular replications happen. Okay, so basically the removal of primers from, let's say looking at the lagging strand here. Okay, it will create basically a shorter, as you can see here, strand. Okay, so let's see what's gonna happen. So the shorter strands here, okay, um, basically are basically what we call telomeres. Okay, so telomeres are the end. They're actually denoted here as orange sections on the chromosomes. Okay, so they're at the end of chromosomes. Okay, um, and these telomeres, they will basically not prevent the shortening of DNA molecules. Okay, however, they do postpone the erosion of genes near the ends of the DNA molecule. So that means we want to have telomeres, right? That because they have a protection uh, benefit. Okay. So it has been proposed that the shortening of telomeres is actually connected to aging. Okay, because telomeres are actually beneficial. If telomeres are shortened, basically due to this is what we're seeing here, right? When many replication cycles, happen, okay, we're shortening the end of the chromosomes, okay, that is connected to aging, okay. So if a chromosome of germ cell becomes shorter in every cell cycle, right, essential genes would eventually be missing, right? That makes sense because genes are fragments of DNA. So if uh, the strand is basically getting shorter every time, we're gonna basically miss or have a dramatic change in genes. Okay, so an enzyme called telomerase catalyzes the lengthening of telomeres in germ cells. So here is the enzyme that actually repairs the telomeres from getting shorter. Okay, so that means if something affects telomerase, okay, that enzyme, that is not a good sign. That's not beneficial. Okay. Okay, so the shortening of telomeres, however, there's also on the other side of the story, might protect cells from cancerous growth, okay, by limiting of the number of cell divisions. So here is the good part of actually shortening those telomeres. So there's a good and a bad part, okay. So there's evidence of telomerase, that enzyme that basically what catalyzes the lengthening of telomeres, right? That its activity in cancer cells, okay, uh, that there's evidence basically that it will allow cancer cells to persist. Why is that? Because it's basically allowing telomeres to stay long. Okay, so if a cell is bad, okay, and the telomerase is basically catalyzing the lengthening of telomeres, right? Basically, it's allowing for the survival of those cells. Then, cancer cells are not going to be able to be stopped. Okay, so that is why, it's telomerase activity. If it's blocked in cancerous cells, that is actually a good thing. We can actually try to stop cancer. Okay. So we already talked about the bacterial chromosome, that it's circular, okay, and we know that eukaryotic chromosomes have linear DNA molecules. They're not circular, okay. So that is how telomeres are created, okay. Okay, we know that the bacterial DNA is in a location called nucleoid, right? So there's no nucleus or nuclear envelope in the bacteria, okay? So that is something that you already know from previous lectures, okay? So let's talk about now the structure of the DNA. So the DNA is wrapped into what we call chromatin. That is the way DNA wraps around, or basically condenses itself, okay? So those strands that we were looking at, right, the double helix strands, they wrap around a protein called histone, okay, as you can see here, okay, and then the DNA strands with histones, we call this nucleosomes, okay, and the whole thing is basically called chromatin, okay. Now, when do chromosomes actually show up like this, in this chromosomal shape? So when chromatins are basically condensed, okay, when does this happen? Only during mitosis, okay? Before that, you're not going to be able to find the DNA in that shape, in the chromosomal shape. You will find it basically, the chromatin is like in spaghetti form all over the place, as you know by now, okay? Okay, so we already talked about how that each one of these is nucleosomes, okay? So it's composed of a DNA wound twice around the core of 8 histones. That is the protein, that circular protein here. Okay. Okay. So you're seeing it here as well, kind of the same thing. Okay. Okay, so I already told you how the chromatin is loosely packed, right? It's uncondensed in its inner phase, so before mitosis, okay? And how it gets condensed into its chromosomal shape in mitosis, okay? Specifically in the prophase, right? Prophase is the beginning phase of mitosis, okay? Okay. That is it for this chapter. So I hope this was good and clear, and we're gonna basically talk about chapter 17 next, which basically talks about how DNA is actually transcribed, and basically after it's transcribed into messenger RNA, how it's translated into proteins. Okay, so stick with me, and I'll see you again in chapter 17. Okay, have a good one, everyone.